Our second presenter is Sandhya Kasivisweswaran, whose presentation is titled To Settle or to Swim, Studying Lifestyle Decisions in Bacteria.
All around us are tiny, single-celled organisms known as bacteria. What if I told you that these seemingly simple organisms have a lifestyle decision to make all the time? In the environment, bacteria can exist in one of two ways. They can either be on their own and free-swimming, or they can live in association with other cells in communities that we call biofilms. You can think of biofilms as little bacterial buildings, where the building themselves are made of a sticky glue-like substance called the matrix that these cells make. Now, as you can imagine, being stuck in a matrix can be protective because it can keep out threats and it can keep out harmful environmental conditions, much like your house protects you from the rain or the snow. But this matrix can also keep out things that the cells actually need, like food or oxygen. To make things worse, cells need to expend a lot of energy to get out of these biofilms. On the other hand, cells can be free-swimming, where they're free to explore, but this means that they're also susceptible to threats like antibiotics. So each of these lifestyles comes with their pros and cons, and the question that we're trying to answer is how do bacteria decide how to live? Should they stay stuck and protected, or free-swimming but at a risk? But why do we care how bacteria live in the environment? Turns out, cells make these very decisions multiple times in the course of infection. The organism that we use to study this is called Vibrio cholera, and it causes the cholera disease in humans. So cells enter the host in biofilms, and this allows them to get through the acidic stomach and into the intestine, where they can then cause infection. Further, when the cell needs to go from one person to the next, cells need to come out of these biofilms and become free-swimming again so that they can move around, making this transition the one that we are particularly interested in understanding. So how do we study this in the lab? Well, we take cells, we put them under a microscope, we color them pretty colors, and we watch, hoping that they'll show us what they're doing. And while studying this very transition, we saw something interesting, which is that while most cells leave these communities and become free-swimming again, they leave some of their friends behind. But why? Why do cells that are all in the same environment respond differently? Well, we've all heard the saying, don't put all your eggs in one basket. Turns out, the bacteria have heard that too, because that's precisely what we think they're doing. They're hedging their bets. While some cells leave and become free-swimming, go on to explore new territory and find fresh food, they leave some of their friends behind to protect the species. We see that when we try to kill all of these cells, the ones that are in the biofilms do better, so they're protected, telling us that in case a threat were to arrive, they can act as insurance, making sure that not all the cells are killed off at once. This clearly shows us that bacteria can present collective behaviors, and that's fascinating to me, so I want to understand it. Through the course of my PhD, I want to think like a bacterium. Thank you.